Hi, my name is Semih Akincilar. I'm a group leader at BioMed X Institute. I hold a PhD in biochemistry from the National University of Singapore and completed my postdoctoral research at the Institute of Molecular Biology, where I investigated cancer progression with a particular focus of telomerase reactivation and telomere-associated inflammation. After several years of focusing on fundamental cancer research, I wanted to move into a new role that would allow me to combine basic research with a more stronger translational impact. Therefore, I joined at 2024 to BioMed X, where I now lead a project focused on developing new strategies to enhance immunogenicity of tumors. My research focuses on understanding how cancer cells create a protective environment around themselves, almost like building a shield to hide from the immune system. Normally, our immune system can recognize and attack harmful cells like cancer cells, but tumors are smart. They change their surroundings, known as tumor microenvironment, to block immune attacks. These tumors are also known as cold tumors, meaning that they have very few cells around them, making it difficult for the body to launch an immune response. Treatments like immune checkpoint inhibitors work much better on hot tumors, which are surrounded by immune cells. However, many patients still have cold tumors that do not benefit from such therapies. Our specific approach is to target the DNA damage repair pathways in cancer cells, and by interfering their ability to fix mistakes, we encourage them to accumulate more errors. This leads them to produce more abnormal proteins called antigens. These new antigens can help the immune system recognize and attack the cancer, effectively turning a cold tumor into a hot one and making immunotherapy more successful. I'm really hopeful that by reshaping the tumor microenvironment, we can open up new treatment options and help more patients benefit from immunotherapy. So in our team, we have 2 postdocs, 1 PhD student, and 2 master students. Due to the complexity of our project, we work in harmony to tackle and identify novel targets to improve the patients' benefits for immunotherapy. Our research program integrates several key pillars, each contributing to a comprehensive strategy for developing an anti-tumor immunity. Using single-cell multi-omics techniques, we leverage clinically relevant response and resistant markers to uncover the most significant interconnected signaling pathways involved in immune modulation. We then perform genome-wide or targeted screens using multiparametric single-cell analysis using spectral flow cytometry to identify targets at the interface of tumor and immune signaling. To understand how these targets work, we apply various molecular biology techniques to identify their binding partners and signaling complexes. In the final step, to validate functional relevance of our targets, we employ various 3D culture models in order to assess alterations in the tumor microenvironment. BioMed X offers a unique hybrid environment that combines the creativity of basic academic research and a strong translational focus thanks to close collaborations with the major pharmaceutical companies. This setup basically allows us to explore fundamental biological questions while always keeping an eye how our discoveries could be translated into real-world medical applications. At BioMed X, every new project starts with a bootcamp, and this is a unique and competitive process where early career researchers from around the world come together and solve a real-world biomedical challenge. For me, it was an intense experience as we had to identify the most effective approach to tackle a complex scientific question within a limited time frame. The process requires harmonizing scientific knowledge with strategic thinking and clear translational vision. For me, the bootcamp was an opportunity to demonstrate leadership under pressure and to align ambitious research ideas with clinical relevance. I strongly recommend everyone to experience this challenging environment. So my goal is to translate our research findings into practical strategies that can improve patient responses to immunotherapy. I think this is highly possible with the unique BioMed X model. Together with our pharma partner, who bring valuable expertise in drug development and clinical translation, we will bridge the gap between laboratory research and real-world applications, ultimately making a meaningful impact for patients.